In last video we did MCQs for biological molecule. Today we are going to do MCQs for enzymes. So enzymes MCQs are starting from here. Which one of the following is called biological catalyst? So catalyst is just enzyme because its definition is already it's a biological catalyst which speed up reaction. So yeah, because clotting factor, osmotic protein, vitamins, these even doesn't suit. So okay, active site of enzyme consists of, so it's consists of only few amino acid, because active site is not a hard or rigid structure which is made up of so many. So this means few amino acid. Yeah, it's the right option. Dash maintains globular structure. So globular structure means now they're making a rigid, like hard structure which would have carried out so many amino acids. So D is the right option here. Bulk of amino acid, because not few means like a little, a few means only like one or two. Many could be right, but bulk means unlimited or like so many to create a globular structure. So D is the particularly right option here. Often it contributes directly to the chemical reaction which bring out catalyst. So it's a cofactor is very important. So I think you will find so many MCQs for the cofactor. So cofactor, I think I could make it here. Cofactor are divided into three parts. First one is I think I could have made it somewhere else. Okay, just okay, cofactor. Wait for a while. In this, simply we have three types. What are these? First one is called as activator, because you will see some other MCQ, so betterly I could do it here. Second one is prosthetic, and third one is called as coenzyme. So the MCQs here was something else, but before that, activator, prosthetic and coenzyme. Activator are simply deattaachable. So here this comes the deattaachable. Prosthetic groups are covalently tightly bound, like these are called as undeattachable or covalent bond. Coenzymes are also deattachable. This means this could substrate could change and they are deattachable. And activator, these are simply inorganic metallic ions like magnesium, manganese. Okay, inorganic metallic ions. Prosthetics are simply kind of organic material, organic. And when we talk about coenzyme, there are also organic material, but it's specifically derived from vitamins like nicotine. Okay, I hope it's clear. So the MCQs here is often it contribute directly to the chemical reaction which bring out, so it's cofactor. Then if non-protein part of the enzyme is covalently bonded, non-protein part is prosthetic group, because holoenzyme, coenzymes, apoenzymes are different things. Prosthetic is a memory based question. I mean, learn it covalent bond which are like non-protein part of the enzyme, that's prosthetic group. If an activated enzyme consisting of polypeptide chain and a cofactor is called as, then it's called as holoenzyme. Holoenzyme now is simply activated enzyme which is consisting of polypeptide chain. Protein, their monomers are amino acids, right? Amino acids, and the chain they, the bond they have in between, like amino acid, the bond that's called as peptide bond. So polypeptide, it means so many chains it become polypeptide. So an activated enzyme which is consisting of polypeptide chain and a cofactor. It also have a cofactor that's called as holoenzyme. Apoenzyme without a cofactor. Coenzyme is simply another thing. Pseudoenzyme, pseudo words means false, not true, like pseudoamoeba, pseudophytes. This means false feeds. Enzymes are sensitive to even a minor change. So enzymes are something, if you change a little pH, temperature, anything in any property in this, it will change its activation, it works. So enzymes are sensitive to even minor change in pH, temperature and substrate concentration. So D is the right option. Dash is a powerful protein digesting enzyme and is capable of destroying cells internal structure, produced in inactive form. So inactive form, pepsin is produced in inactive form. Okay, but here pepsin is produced in inactive form, but we activate it by pepsinogen by adding HCl, and this happens in the lumen of stomach. like with the lining, with the mucus of the stomach. Okay, so B is the right option, because pepsin is a powerful protein, but pepsinogen is not an inactive form. So this means A is the wrong. Pepsin and trypsin, these are different thing. Trypsin and pepsin, again this is wrong, so B is the right option. After the formation of product, it is released unaltered, thus can be used as substrate. So anything, I think substrate and enzyme, not enzyme but substrate, can be used again. It's just formation of product is done. For suppose if we need this product, this is just now done for the formation of product. Now it's released and unaltered, so this means we can reuse substrate. Electron, not electron, sorry, enzyme plus substrate, then this reversible reaction become enzymes and substrate, not enzymes and product, because after this chain it become this thing, not this one, even EI inhibitor, no. Okay, so B is the right option. Enzymes involve some metabolic pathway normally present together in precise order, just learn it. The charge, and because randomly, no, they have a particular series, descending order, yes, reverse order because it's a reversible type. So precise order, this means they're not elongated and not so elaborated, right? So charge and shape of the active site of the enzyme is formed by some amino acid in the polypeptide chain. Okay, the reaction between dash activates the catalytic site of enzyme. So the right option is C. The reaction between activate the catalytic site, its substrate and the binding site, those activate. If you see enzyme like this, is simply a shape. The site where any substrate get attached, that's called as active site. So substrate which get attached, for suppose we have to experiment that okay, even here we have a, so I'm raising this thing, substrate and binding site of the active site. Now according to this model, dash is a rigid structure. So we have two models. First one, lock and key model. Second, induced fit model. Lock and key model was given by Koshland, and induced fit model was given by Emil Fischer. Emil Fischer gave experiment, like did experiment in 1998, if I'm not wrong. This was 1959. Okay, these both experiments are quite opposite. Lock and key model says if enzyme is present, so not any, for lock and key, this even tells if there's a lock, we need a particular key to unlock it, right? So same way this says, no any, if this is a body, no any enzyme, no any substrate could bind. Okay, we need a particular thing. So if shape is like this, so we need another substrate which of this shape, not of the triangle shape now. Okay, so like this way lock and key models, according to this model, which is rigid structure. So yeah, lock and key model is a rigid structure. Now comes according to this not rigid structure, now it's induced fit model. Why? Lock and key model are simply given already the opposite. Emil Fischer was the scientist who discovered, so this is already elastic model, is also not this thing. Okay, so induced fit model is the right option. Then comes the function. The function specific of every enzyme is a consequence of a specific chemistry and configuration. Why? Because they're very sensitive to pH, temperature, so any change, so this means they're quite specified to the chemistry of the bonding, charge, reaction, energy, everything. So specific. The rate of reaction depend directly on the amount of enzyme provided the substrate. So substrate, like initially reaction begins, product gets continuously, and it become like unlimited with the speedy reaction. So A is the right option. By increasing enzyme molecule, dash will convert substrate molecule into product in the given period of time, so it become more active site, it gets more active sites if the enzyme concentration is kept constant. Sorry, by increasing substrate concentration, the rate of reaction is, I think, increase for specific time. Why is it so? Enzyme concentration is kept constant. Now enzyme concentration is constant, but we increase the substrate concentration. Enzyme is constant, we keep substrate concentration increasing, so the rate of reaction until the substrate is provided, until the concentration is kept constant, the reaction will increase for a specific time, not forever, never increase, no it's wrong, decrease, no it's not decrease, but the right option is D, increase for specific time. Yes, rate of enzyme controlled reaction may increase within temperature up to optimum level, so that's required even for a human body. Our body works for the 37 Celsius, which is very important. After that or before, like lower or below than that, our body does not work, so they are very particular to pH, temperature, everything. Chemical reaction are accelerated at high temperature because heat provides is the activation energy. Just because of that, because heat lower, no, when activation energy is there, so I mean heat lowers the activation energy, no, it's wrong. Heat lowers the kinetic energy, no, but heat provides activation energy to speed up the reaction. It makes the reactor, no. When the reactant are more quickly and chance of their collision with each other increase as a result, it become increase initiality. Inhibitors can be divided into two basic types, okay. So we have two types of inhibitor, reversible and irreversible. Second, the next question is, I'm sorry. Next question is they alter the structure of the enzymes in such a way that they, even if gene fails to take place temporarily, so it's non-competitive inhibitor. This means they are reversible reaction. They particularly need to start up a reaction. Reversible, this means that could be sometimes working on both sides. Pick up the product. So pick up the product, this means formic acid is a product. How? Because when you make a reaction like two hydrogen get reacts with two hydrogen combined form a succinic acid, and that reaction get catalyzed from succinic acid and dehydrogenase enzyme, so the product become formic acid. Succinic acid, sorry, this one, okay. Succinic acid dehydrogenase plus malonic acid, when this type of anything happen, this mean when you say succinic and dehydrogenase together, this mean and malonic acid, this mean no reaction could succeed because enzymes get blocked by this reaction. Which of the following is reused? So enzyme and coenzymes are already like reused, okay. uh, some enzymes are potentially damaging if they are manufactured in the active form. Active form, pepsin is an active form, pepsinogen is an inactive form. So it get activated in like by adding of the reaction of HCl in the lumen of stomach and some lining or the mucus of the stomach as well. Which of the following product will control their pathway through feedback? So which of the following product will control this pathway through feedback activation? So it's E, okay. Accumulation of E will control above pathway through. So here feedback inhibition occurs, okay. So here feedback inhibition is simply a process when there's no need to create or action to be done. So inhibit means to stop. So inhibitor are like something, some reaction, like these are very important thing in the enzyme chapter, enzyme thing, because if no reaction is supposed to be need, like no reaction, no change in our body is required, and still they are continuously making it, so the feedback inhibition will order them to stop their reaction to inhibit them. And if we do opposite of the deficiency of E, this means E is simply a product, end product, okay. E, if it become deficient of this, so it become feedback activation. So B is the right option. Induced fit model was proposed by Koshland, and lock and key model was proposed by Emil Fischer. Salivary amylase work best at the 7.2, or I think not 7.2, yes, my bad, not 7.2, because they work nearly to the neutral pH. It's like 6.80, because if you see a scale, 7 is the neutral and they're just a little back to that. So and 7.22, these are the activation, like first of all pepsin work at the pH of 2, and 8.5 is so acid, so alkaline. 7.20 is also so much like, okay. Then comes malonic acid. It's a competitive inhibitor of succinic dehydrogenase. We did here it, okay, sorry, we skipped one MCQ. The optimum pH for action of pancreatic lipase. So pancreatic lipase work at like 9 pH, which is too much, but yeah, it works like that. Water has a pH which is 7, quite neutral. 2 is the pH of pepsin. Then comes, it causes degeneration of globular structure of enzyme. Then because of the extreme pH, denaturation occurs when, this means they started denaturing, and that action happens when the pH is extreme pH, like so much, like we are boiling or anything like that, extreme pH, then this type of reaction occurs. A chemical, sorry, okay, any molecule that increase rate of chemical reaction without being used up that reaction. So catalyst simply which speed up reaction even they are not used themselves. For suppose we say enzymes are biological catalysts which are used. okay, but they like here it's written what is something which speed up reaction instead of being used. So it's catalyst, apoenzymes, coenzymes, activator, simply something else. Activator are deattageable, some organic metallic ions. Apoenzymes, which does not have a cofactor. Coenzyme, which is simply deattageable but have organic dry material like from vitamins like nicotine and those. So yeah, these are enzymes MCQs from all the competitive exams. I hope you got everything. Thank you so much for watching.